Good morning. So much energy. Okay, so some of you guys asked me for my contact information. It is found in the first slide of every lecture that I put up, so it's easily accessible. My regular office hours are Fridays at 3 p.m., but for the next 2 weeks, due to my other teaching and symposium, I have to change these hours, so I put an announcement at the NYU classes website, and also the hours are here on the slide. So if you want to stop by, come by at these times. If you can't make it, you're working or etc., then email me to make an appointment, okay? All right. So today we're going to continue on the eukaryotic transcription. It's going to be a lot of information dense lecture. Basically just giving you the information about the basics of eukaryotic transcription. And then after you guys learn a little bit more about molecular biology techniques, etc., in the next few lectures with Professor Siegel, we're going to go through a lot of molecular biology techniques and experiments that allowed us to gather the information that is presented in this lecture. Okay? So hang in there and try to understand this information. So we talked about transcription and the transcription machinery in prokaryotes, and the eukaryotes. This process is pretty much conserved, except there is more complex problems and issues that eukaryotes face, and that's going to make it a little bit more complex. So the machinery is well conserved. So if you look at the bacterial core RNA polymerase enzymes, we talked about the beta prime subunits and alpha subunits and omegas. If you look at archaea, you find the homologs of each of these subunits. And when you look at the eukaryotic RNA polymerases, 3 of which are the major RNA polymerases that are shown here, RNA pol 1, pol 2, and pol 3, what you can find is you can find the homologs for each of these enzyme subunits, plus there are more subunits that are involved in the transcription process. Okay, so it's more complex. It has a little bit, it has more proteins that do the job and additional jobs. All right. So how did people actually first recognize that there are different RNA polymerases in eukaryotes? Is that they did a biochemistry experiment where they looked at the ability of protein fractions that are coming out of column chromatography and see if they can, if these fractions are capable of transcribing DNA, and also used a mushroom toxin, alpha-amanitin, to see if those polymerases are sensitive. Okay, what do I mean by that? You guys have learned about columns in biochemistry, or no, you haven't taken biochemistry, but you learned about column fractionation of proteins, correct? Is that right? Yeah, so basically the idea is very simple. What you do is you prepare a matrix, a gel matrix. It could be gel or it could be something else, right? Let's say this is a size exclusion chromatography, so it contains a lot of these beads. And what you do is you take your cells, you break them up and make a protein mixture, okay? And then what you do is you put this protein mixture on top of the column. So, and you wait, okay? So the speed by which the proteins are going to travel through all this matrix is going to be determined by their size. So what you do is you put a little bit of a little tube under this, okay? And then wait for these protein fractions to come out. So the first fraction that will come out is going to contain proteins that are going to be smaller, right? Because they're going to reach the end of the column first, because they can wiggle around really easy, right? And then you're going to wait a little bit. You're going to remove this fraction, put it here, right? And then what you're going to do is you're going to put your next tube in under here, and collect that, and it's going to be a little bit bigger proteins will come out now, right? And now you're going to put that fraction here, etc., etc. And so the proteins that are larger or multiple proteins that make very stable contacts with each other, forming protein complexes, large protein complexes, are going to come out at the later fractions. Does this make sense?
I thought that the beads had pores in them, so that smaller particles got stuck in the beads and came out last, not first.
Depends on the type of the column chromatography. There are different matrices that you can use. The matrices that will allow smaller proteins to come out. There will be matrices that allow larger proteins come out first. There will be matrices that allows positively charged proteins come out first, etc., etc. So there is not just one column chromatography. There's multiple column chromatographies that will separate the proteins based on different different properties. Okay. So you guys learned with Professor Siegel the gel chromatography, correct? Yeah. Okay. Thank you. All right. So basically, what the idea is to separate the proteins based on their size or charge or something, etc. Right. So the idea is to separate proteins and protein complexes. So what we're seeing here is that we're seeing each fraction that is tested in this assay. Okay, so the first thing that the researchers are doing is they're plotting the total protein amount per fraction, right? In this fractionation, it appears that a lot of the proteins, most of the proteins are coming out first, right? And then there are few proteins that are coming out later. Does that make sense? So that's the black line here. So most of the proteins are coming in earlier fractions than later fractions. And then what they do is they take each of these fractions, they put a DNA template in there, they put some nucleotides, and then they see whether these fractions can produce any RNA. And then they plot the RNA synthesis in the pink. Okay? So what you can see is all of these proteins that are coming out first, are they capable of all of these fractions? Do they contain proteins that are capable of synthesizing RNA? No, right? So you don't see any RNA synthesis. But then what you can see is that there are some proteins in these fractions that are capable of producing a lot of RNA. And there's another fraction, they look like peaks, right? Because the fractionation is not perfectly separating the proteins. So you find a protein each protein is going to be found in several fractions, but it's going to be shown like a peak here, right? So a lot of the pole 2 is found in this fraction, but a little bit of it comes out in the previous, and a little bit of it comes in a little bit the later fraction. Okay, so that's why you see kind of this triangle type of shape. And then you kind of see here there's another peak here. At the time, they didn't know that was a different polymerase, but when they did an alpha-amanitin sensitivity assay, this is basically a mushroom toxin that was known to inhibit some RNA polymerases. So what they did is they took all these fractions, they set up the RNA synthesis assay again, and to each of them they put a little bit of alpha-amanitin. Okay, and then they plot the RNA synthesis amount with after addition of alpha-amanitin in this kind of blue shaded data. Okay, so what you can see here is pole 1, the fractions that are synthesizing the RNA in here, are they sensitive to alpha-amanitin? No, right? Because the blue line is just as high as the pink line. Right? So the RNA synthesis is going fine, even in the presence of alpha-amanitin. Does that make sense? The same is true for the last few fractions that were shown here that is capable of synthesizing RNA. But this fraction here that synthesizes RNA is very sensitive, right? So if you add alpha-amanitin, which is shown in the shaded blue, you no longer see any RNA synthesis at all, right?
Why do we need 3 different polymerases if each of them can do the job on their own?
Each of them are specialized to do a different job. it's a good question, why, but it's, and we can speculate, right? But in biology, we can't really answer whys as easily as how or what, right? So each of these polymerases are basically specializing to transcribe a different set of genes. So RNA polymerase 1 in eukaryotes is specialized to transcribe rRNA genes, so it's found in the rDNA, right? And then RNA polymerase 2 is transcribing mRNAs. So these are messenger RNAs that are going to be used for translation to make the proteins, right? So these are your typical mRNAs with the poly A tails and spliced and etc. And then RNA polymerase 3 is specialized to mostly transcribe tRNAs and small non-coding nuclear RNAs, okay? So they are specialized for different transcribing different genes. And at the end of the lecture, you're going to also see that they also have all these initiation subunits and additional subunits that help them guide towards the promoters of these specialized genes. Okay? All right. So how do these proteins actually look like? The prokaryotic RNA polymerase looks something like this. I always say it looks like a hand to me, right? And they have the beta prime subunits, alpha subunits, and omega subunit here. And the yeast RNA polymerase and the human RNA polymerase is very similar to each other. And structurally, it looks also very similar to the bacterial RNA polymerase, right? But there are certain features that distinguish it. One is that the eukaryotic RNA polymerase has a lot more additional subunits that associate with these core subunits that you also find in the prokaryotes. And in some eukaryotes, but not all, there is the C-terminal domain of the RNA polymerase that sticks out here, which contains multiple repeats of the same amino acids. Okay, so that's the C-terminal domain repeat. So that's the C-terminal end of the RNA polymerase, large RNA polymerase subunit sticking out, and you have the same amino acid repeated several times. Okay, in humans, this is about in different eukaryotes, the number of repeats differ, but you typically find between 26 to 52 repeats of the same amino acids. Okay, so this C-terminal domain has an interesting sequence in that if you analyze this C-terminal domain repeats, what you see is that several amino acid residues here are phosphorylated. So what you can see here is that, I can't remove those. There is a phosphorylation of serine 2, so serine 2 for phosphorylation. There's a threonine phosphorylation, serine 5 phosphorylation, and serine 7 phosphorylation. So the C-terminal domain has several phosphorylations on it. So the researchers asked then, what is the function of these phosphorylations, right? And this was asked decades before, where you could not actually look at this problem very easily, because they wanted to know if these phosphorylations are important for transcription or not. Okay? And to ask such an experiment and to answer this experiment, what they need to do is they need to map phosphorylated or unphosphorylated of the RNA polymerase in the genome and see if the genomic regions that are transcribed is associated with the phosphorylated form or non-phosphorylated form, right? And it's difficult to see which parts of the genome is phosphorylated or not. So what they used is a system that is found in Drosophila. And these are what we call the polytene chromosomes. So Drosophila has these chromosomes. We talked about DNA packaging, right? In interphase, do you actually see separate, nicely condensed chromosomes? No, right? They're kind of all in the nucleus, kind of like untangled. There's some more dense parts, there's some less dense parts, but you don't know exactly what genes are in the less dense euchromatic region and what genes are exactly in the heterochromatic regions, right? You actually don't know. They're all mixed up together. in mitosis, when you see those nice condensed strings, the fibers, right? In polyethylene chromosomes, you actually can see the chromosomes, individual chromosomes, and you see the linearity of the chromosomes shown here, right? And then you can stain these chromosomes with a dye, and that shows this banding pattern. It's very similar to GEMSA staining of the mitotic chromosomes, right? Based on this banding pattern, before the genomics era, before we could sequence anything, the researchers were capable of telling which chromosome is which, right? And based on the pattern, they would know, okay, this is the start of chromosome 2, and this is the end of chromosome 2, etc., etc. So how do these polyethylene chromosomes arise? They arise due to a very interesting phenomenon where the DNA replicates. So you make many copies of the chromosomes, so all these sister chromatids, but you don't segregate them, okay? So each of these chromosomes basically have lots of chromatids that are bundled together. So it's kind of like a cable with lots of cables bundled together, and you can now see them, all right? The advantage of using these interface chromosomes that are visible is that they also knew that certain places in these bands contained genes that were responsive to, for example, heat shock, right? So in our genomes and the Drosophila genome, when we are in a stressful environment, when our cells are in a heated environment, there are some genes that need to be expressed to take care of the proteins, so make sure that everything goes well in the cell. So some of these are protein chaperones. The proteins, when they're heated, they tend to kind of like start denaturing, right? So these proteins kind of fight against that. So these heat shock inducible genes were found in these banding regions, and when they're heated, they kind of puff. And any ideas why these regions might puff when you have heat shock and you have genes here that needs to be expressed in the heat shock? Any ideas? Why do you see the puffing?
Because the chromatids decondense, so transcription.
Exactly. So these bundles are together very tight, packed very tightly, right? And when they need to be transcribed at a region, that bundling opens up. You need to open the chromatin to make it more accessible so those genes can be transcribed. So by looking at this, what you can see is that the genes here are being transcribed, and the genes here are not being transcribed. Okay? So that's what the information they had. Then they said, okay, let's now make antibodies to these C-terminal repeat regions and make two different antibodies. One of these antibodies they're going to make in goats, okay? And these antibodies are going to recognize the antigen, so the C-terminal peptide, that's completely unphosphorylated. So that's 7 amino acids, and none of them are phosphorylated. And then they make an antibody in rabbits, right? And then so that antibody, they use peptides that are phosphorylated. And the way that you make these antibodies is pretty crude. What you do is you make your peptide chemically or otherwise. You phosphorylate them, right? And now you go and inject into the rabbit. The rabbit, that's a strange protein. It's a strange antigen, right? And it produces a lot of antibodies and releases into its serum. So 6 months later, you bleed the rabbit. You take the blood, right? You spin down all the blood cells, and then you can purify the antibody. And then a lot of those antibodies are going to be against the phosphorylated C-terminal peptide that you injected 6 months earlier. Does that make sense? So you do this in goats and you do this in rabbits. Okay. So next what you do is you use a secondary antibody that recognizes the goat heavy chain, right? So you're going to have a secondary antibody that's recognizing only the goat heavy chain, and you're going to to attach a fluorophore to this, that's going to show up as green. And then you do the same for the rabbit antibody, but instead of green, you're going to add a red fluorophore, right? And now what you're going to do is you're going to take your polythene chromosomes, put it on a microscope slide. You're going to take your antibodies, you're going to put onto your chromosomes. You're going to wait until the antibodies can find the targets, right? So they're going to find the phosphorylated or non-phosphorylated CTDs on there, right? Now you're going to wash the antibodies that are not bound. You're going to take that slide and put it under the microscope, and what you're going to see is you're going to see the non-phosphorylated form of RNA pol 2 found at genes that are not transcribed or very lowly transcribed, and then the phosphorylated form of the RNA polymerase 2 is found at these puffs, suggesting that phosphorylation is correlated with transcription. Okay? Make sense? Okay, so in fact, the different types of phosphorylations or modifications that you see at the C-terminal are correlated with different stages of transcription. So what's shown here is that at the pre-initiating stage, where the RNA pol 2 binds to promoter but is not phosphorylated, is basically the polymerase that's associated with initiation. And then once it starts initiation, the C-terminal CTD repeats get phosphorylated as serine 5 by a subunit in the TF2H. And then when the polymerase starts elongating, the phosphorylation also gets added to serine 2. And then towards the end of the transcription stages, where the terminating polymerase, what you see is a serine 2 and serine 7 phosphorylation. All right. So you see different phosphorylations associating with different transcription stage. So that was the difference between how the eukaryotic machinery is a little bit more complex than the prokaryotic machinery. What about the gene regulatory elements? Again, the eukaryotic gene regulatory elements, you have more of them, and they're a little bit more complex than prokaryotic elements. Okay? So in prokaryotes, what you have typically is you have an open reading frame, and right upstream of it is the promoter, right? And at this promoter, you have these consensus sequence at -35 and -10, bound by the sigma factor. You might have an operator sequence that's bound by the repressor, right? And you might have other sequences that are nearby that are going to be bound by activators. So that's the typical simple organization of the prokaryotic regulatory elements. In eukaryotes, in a small eukaryote like yeast, as Cerevisiae, what you find is you find the genes, and you find a consensus sequence, that's the Tata box, right upstream of the gene in the promoter region. And then you can find enhancers that are bound by transcription factors that control the activity of this promoter. And in yeast, they can be called upstream activating sequences like UAS or upstream repressing sequences like URS. Okay? In mammalian genes, the picture gets a little bit more complicated. So what you have here is you have your typical coding region, right? But this time the coding region is divided into exons and introns. And upstream of that, you have the Tata box that is located at a stereotypic distance from the transcription start site. And then you have additional DNA sequences here shown in red. These are called promoter proximal elements because they're close to the promoter. They are typically involved in tethering the transcription initiation factors to the promoters. So this is the promoter of the gene. And then you have these enhancers. And for a given gene, you might have multiple enhancers. So enhancers are these little genomic segments that can be far away from the promoter but control the activity of these promoters. These enhancers can be found upstream of genes, a few kb, or a lot more. So it can actually even skip a gene. It can be very far away. It can be also downstream of genes, and it can be found in the introns of genes. Okay? So these enhancers can be everywhere. And what they do is they control the tissue-specific and temporal expression of this gene. And we're going to talk about how we can find these enhancers, how we can visualize them in a few lectures from now when we learn about reporters. All right. In mammalians, the TATA-containing promoter structure is not the only structure that you can see. There's also another a class of genes which contain CPG islands. Okay, so in these genes you see again exon and intron, etc. And then they have some promoter proximal elements. They don't contain the exact TATA sequences. So these are this TATA sequence is TA, etc. So they don't contain these TATA sequences. And they have less precise transcription initiation. So instead of one site where the transcription initiates, you can have multiple nucleotides where the transcription initiates. And interestingly, the transcription can initiate in both directions, although when it's initiated in the correct direction, it will produce these RNAs that are long and produce the mRNA. When they are starting in the other direction, they typically produce short RNAs that get degraded. So they don't make up these long, stable mRNAs. Okay? All right. So this is about 60% of the promoters. So our genes contain these CPG island promoters. A lot of our genes contain these CPG island promoters. What do I mean by CPG island? Okay? This is basically the description of what a CPG island means. So, if you look at the genome sequence, you're going to you're not going to find a lot of C's next to these. CPG essentially is the same thing with a C nucleotide right next to a G nucleotide. P just stands for the phosphodiester bond, okay? CG, that's what it is. When you look at the genome, you don't see a lot of CG, except some regions where you find a lot elevated CG levels, so you find more than what you would see on average, and frequently less methylation of the C. We talked about in human genome, a lot of our cytosines being methylated, right? We said a lot of the cytosines are methylated. So in vertebrates, Cs are methylated. Most of the Cs are methylated, except in these islands. You see a lot less C methylation. All right. So why do we have a depletion of these CGs in our genome? Okay, so most of the Cs are methylated, and sometimes these methylation can cause mutations, so that over millions of years, the amount of CG sequences are going to reduce. So does anyone remember why CPG methylation may be mutagenic and why the C can be converted into a T? Do you guys remember that process? You were here for the repair lecture. What can happen to DNA?
Can't the C shift to the T and that cancels the mutation, and if it's not checked, it still goes through?
It does. So if the C can turn into a T, if it doesn't get repaired, so it goes through a next replication cycle, it can turn into an inherited T, right? So C converting to T. What was the chemical basis of that? Deamination. So when cytosines are methylated and gets deaminated, it turns into a T. And then what you will have is you will have a T-G mismatch. If the repair enzymes catch it, fine. It's going to go back to a CG. If they don't, then it's going to turn into a TA in one of the sister chromatids, right? So you can go back to that lecture and find out how that occurs. And over time, that occurring repeatedly, a lot of the Cs are going to be Ts. All right. So how do these basal promoter elements actually look like? We talked about how you can determine consensus sequences and how these consensus sequences were found at the minus 35 and minus 10 region of the promoter in prokaryotes. In eukaryotes, similarly, if you take eukaryotic promoters, line them up by their transcription start site, and look for sequences that seem to be conserved, you will find these sequences. For example, you'll find the TF2B recognition element. TF2B is the initiation protein, just like the sigma factor, similar to sigma factor. So this is called the BRE element. And here the consensus sequence is written in a way that's a little bit different from a motif logo, right? So what it says is you can find a GGG or you can find a CCA followed by a CGCC sequence. So the consensus, what this way of expressing a consensus sequence is telling you is that in some genes you find GGG, CG, CC, and in some other genes you find CCA, CG, CC, okay? And similarly, the Tata box looks like this. When I say Tata box, it's named because the sequences that you find most are called Ta, right? Tata followed by a strings of A's and T's. In Drosophila and mammals, you also find this sequence here that is similar, this consensus sequence, and there are some promoter proximal elements that are close to the promoter transcription initiation site, but instead of being right upstream, they're actually a little bit downstream, so at plus nucleotides, etc. One of them is called the downstream core promoter element. What do these guys do? These guys make sequence-specific contacts with general transcription initiation proteins. Okay? These general transcription initiation proteins are going to recruit the RNA polymerase to the promoters. How does that occur? So this is for a Tata box containing gene. In the first step, what you're trying to do is in this big genome, you want to target the RNA polymerase to the promoter. You don't want the RNA polymerase going somewhere else. Okay? There are a lot of proteins specialized for this function. They're called the initiation factors. One of these proteins is the Tata binding protein called TBP. And what it does is it recognizes that Tata sequence. And along with another protein complex called TF2D, they bind to the Tata sequence, and the way that it binds, it bends the DNA. So it makes these interactions with the Tata sequence, and it kind of bends the DNA. All right, so once that structure is formed, there are other proteins called TF2A and TF2B. They recognize the Tata binding protein that is bound to the DNA, and that molecular conformation helps it to get recruited and attaches itself. And once you have that, you have now this is the structure that can recruit the RNA polymerase that is also complex with another initiation factor called TF2F. So this RNA polymerase is going to make molecular interactions with the Tata, TBP, and what it is going to do is that interaction is going to position the RNA polymerase downstream where the transcription needs to start. Okay? Make sense? What happens if you reverted the Tata sequence? What would you find? It would go the other way, right? Because these interactions are directional. So the RNA polymerase can only recognize this side of the TBP, right? So if you reverse the sequence, TBP would be making contacts with the Tata sequence the other way, and then the polymerase will be facing the other way, okay? Next, what happens? You have another initiation factor called TF2E. What it does is it interacts with the RNA polymerase and creates a site where another initiation complex called TF2H with 10 subunits can come and bind to this pre-initiation complex. So all of these different initiation factors, along with RNA polymerase that is bound to the promoter, is called the initiation complex. Okay, so that's the complex that's going to initiate transcription. So TF2H has a helicase activity. One of its subunits is a helicase, and that's going to use ATP to open up the DNA double helix to form an open complex. So far, until TF2H, it was a closed complex, and then when TH2H comes in, it opens up. Anybody remember this enzyme? Yeah, we learned about this TF2H somewhere else. Recall. That's how you learn. In one of the repair lectures. Very good. Nucleotide excision repair actually uses this enzyme to open up the DNA for the repair, right? So same set of proteins are actually used for DNA repair as well, because one of the things it can do is the helicase activity, opening up the DNA. All right, so what happens next? What happens is that TF2H has another subunit that's a kinase. It phosphorylates the serine 5 at the C-terminal tail of the C-terminal repeat. And that serine 5 phosphorylation is basically a docking site for 5' capping enzymes. So these are the enzymes that are going to cap the 5' end of the mRNA, right? And what they're doing is they're getting recruited to the promoter. Again, you got to recruit the protein to where it's supposed to do its job, and that recruitment mechanism is through the phosphorylation of the CTD. Okay? So when it's 5' CTD, finds that. All right. So once the promoter is the RNA polymerase is released from the promoter, so we call this escapes from the promoter and starts initiating transcription. What happens is that all of these general factors are released from the promoter. They no longer migrate with the RNA polymerase, except that TBP usually stays bound to the promoter, the TATA. And that's why we call these initiation factors, because they're important for initiation, but they're not important for elongation. They're just released. So most pre-initiation proteins disassemble as the POL2 starts elongating. So which factors in prokaryotes is an initiation factor? Which factor disassembles from the RNA polymerase when it starts transcribing? Very good. It's sigma factor, right? So it stays. It doesn't go with the polymerase. Great. All right, next what I'm going to do is I'm going to show you a movie that was a result of decades of work with multiple people, and what they've done is they really wanted to understand this, what we call molecular machinery. How does this complex actually transcribe the DNA, okay? And what they've done is they painstakingly took every subunit, every initiation subunit, every polymerase subunit, and they crystallized. So they can create these crystal structures, and they can see how it forms when you add DNA, what happens when you add nucleotides, when it happens when you have a mismatch, etc., etc. And then they came up with this very nice movie a few years ago. Okay, so what you can see here is you can see the RNA polymerase first, and it's facing like this, so the hand and the thumb is looking up. And it's showing you the clamp domain and the cleft, and this is the active center with the magnesium. Okay, so it complexes with some of the pre-initiation proteins, so it's binding here, and you can see this. And once this enzyme is formed, it can actually recognize the TBP that's bound to the DNA, shown here in blue, and here's the TBP, and that's the TF2B. And what the TF2B does is it makes contacts with the RNA polymerase so that this RNA polymerase is positioned perfectly right next to the DNA that's going to start transcribing. Okay? So these are the different features of the TF2B protein that's making those contacts. So next you have TF2H, you have opening of the DNA, forming that open complex. And the active site is going to start transcribing. So you're going to have an initially transcribing complex at the promoter. So that's the RNA shown in red, and that's the NTP coming in orange. So the NTP is going to be added to the growing chain of RNA. So at first, this complex actually stays bound to the promoter through this interaction with TBP, etc. It doesn't move. But what it does is, as it adds more RNA, it basically scrunches the DNA out like this. Okay, so you have an initial scrunching of the DNA. Once the polymerase escapes the promoter, then the structure looks a little bit different. You no longer make contact with the TBP, etc. So here is your open DNA. So this is your RNA is going to exit from this side. And what it's going to do is it's going to make more protein-protein interactions with some elongation factors shown in orange. So these factors don't bind to the RNA polymerase at the initiation stage. They bind at the elongation stage. And the reason why they can't bind at the initiation stage is their binding sites are actually covered with initiation factors. Now what we're doing is zooming into the catalytic site. Okay? So the catalytic site has this bridge, has this alpha helix, and then another bridge helix, and some other alpha helices that are called the trigger loop. And it'll be clear in a second. And you have magnesium here at the catalytic site, and this is the growing RNA. Okay. So the first thing what's going to happen is that another magnesium is going to take the RNA ribonucleotide. And this ribonucleotide is going to sit at the catalytic site, and it gets pushed by that trigger loop. And the enzyme is going to catalyze the phosphodiester bond, and that pyrophosphate is released. The release of pyrophosphates basically pushes back the bridge helix and the trigger loop out. And then what is going to happen is the DNA is going to translocate. And then what you have is the next nucleotide template coming in. Now you're going to start again. You're going to bring this pre-insertion conformation that's going to fit that new nucleotide coming in. The enzyme is going to catalyze that bond, right? Form that bond. And then pyrophosphate gets released, and that changes the conformation of the helices again, and that's going to trigger the translocation, and a new nucleotide is going to be coming in and pushed into the catalytic site. And here's RNA polymerase, how alpha amanitin inhibits polymerase. What it does is it inhibits that translocation, so RNA polymerase gets stuck in that process. Okay. All right. So here's what can happen when there's a mismatch. So when the RNA polymerase accidentally adds a different nucleotide, it can actually backtrack, and another part of the enzyme can cleave this previously made bond and remove that mismatch. Okay? Sometimes what that causes the polymerase to do is it backtracks on the template a lot more. So what it is going to do is it's going to come backwards. So the RNA, that's newly made RNA, is going to come out, and it's going to start exiting at another domain of the RNA polymerase. And this domain of the RNA polymerase is going to this is going to arrest. So it's going to backtrack and arrest when there is one of these mismatch problems. That arrested structure is going to be recognized by another protein called TF2S. And what it is going to do is it's going to come and make an endolytic cleavage. So it's going to cleave the RNA at the phosphate backbone. And when that happens, that RNA is removed. This protein complex is going to rescue. So what it's going to do is it's going to switch to being the normal elongating complex and continue elongation. Okay? And that occurs when RNA polymerase encounters with problems, etc. Isn't that beautiful, right? This is what you would call molecular machinery. It's machinery made out of proteins doing something that's amazing, right? Reading your genome. All right. So we said that, man, seriously. Okay, don't sign. The computer was so excited it couldn't handle it. All right. So eukaryotes have a lot of, also have other things to deal with. Okay, in prokaryotes there is no histones where the DNA is wrapped around, but in eukaryotes the DNA is wrapped around the histone proteins forming nucleosomes. The RNA polymerase can't open the DNA and start inserting stuff, etc., when the DNA is nucleosomal, right? So what that means is these nucleosomes need to be removed. Also, the initiation proteins cannot recognize and bind to those initiation elements if those elements are also wrapped around the nucleus, okay? So the nucleosomes prevent the accessibility. So what has to happen is that you have to remove these nucleosomes from the promoters and at the initiation site so that you can start recruiting the pre-initiation complex and the RNA polymerase. And how does that occur? So that occurs through transcription activators. So these transcription factors in eukaryotes, you learned this in Principles of Biology. This is transcription factors. This is one in yeast called GCN4. It has two domains. One is a DNA binding domain, and this DNA binding domain is recognizing and binding to the sequences at the enhancer of the yeast gene called upstream activating sequences. And there's another domain in this protein called activation domain. And what this activation domain does is it physically interacts with a protein complex that contains a subunit called GCN5. This protein complex in yeast is called SAGA. It's a chromatin modifying protein complex because it contains a histone acetyltransferase, a HAT. And the HAT is GCN5. And what this HAT does is it acetylates the tails of the histones in the nucleosomes nearby. Okay? When you have these acetylation, we talked about the acetylation kind of loosening up a little bit the nucleosome and chromatin structure and opening up a little bit, right? But that kind of opening up is actually not enough to remove these histones and unravel the nucleosome. You actually need another complex. So that complex is called switch SNF, which we call an ATP-dependent chromatin remodeling complex. And this complex can actually physically remove the histones from the DNA, okay? So freeing the DNA. And the way that it gets recruited to the promoters or acetylated promoters is it contains a subunit that can bind and recognize and bind acetylated histone tails, right? We talked about how acetylated histone modifications can act like binding sites for certain proteins. That's what's going on here. So these reader proteins contain this domain, a reader domain called bromodomain, that bind to acetylated histone tails. So they're found in switch SNF. They're also found, interestingly, in a general initiation factor called TF2D. So you can also, when you acetylate the nucleosomes, you can also start calling in this initiation factor saying, get ready, here's some acetylated promoters to come and start transcription. All right. So then the nucleosomes get removed. Everybody comes in, like we talked about with the initiation complex, and you can start the transcription. So this same mechanism can also be used in eukaryote to repress transcription. So to initiate transcription, you need to kick out the nucleosomes, right? then what you can do to repress a gene is you can make the promoter wrap it around with the histones and form nucleosomes. How does that happen? So this is a gene here. At this time it has an upstream repressive sequence, right, URS. And that sequence is recognized by another transcription factor called UME6, and this time this is a repressor, okay? And again, the organization of this transcription factor is exactly the same. What you have is you have the DNA binding domain binding to here, and it has another domain, and this time it's called a repressive domain because it makes interactions with a protein complex that contains a subunit that deacetylase histone tails. And this deacetylase is a histone deacetylase. The shorthand is HDAC. And once this repressive complex comes in, it can remove the acetyl groups from the nearby nucleosomes, right? And then the nucleosomes are going to be more stable when they are deacetylated. And so the SAGA complex, so that the TF2D doesn't bind, so that switch SNF doesn't bind, so this promoter stays nucleosomal, okay? And so these chromatin modifying complexes, we also call them co-activators or co-repressors because they act with transcription factors to activate or repress transcription. So in this case, this RPD3 HDAC complex is called a co-repressor. All right, so there are a lot of readers and writers and etc. in this case, right? So in this system, in the previous slide and this slide, what protein is the writer of acetylation? Just look at your notes and see which enzyme is the writer. GCN5, because that's the acetylase, right? Which proteins are the readers? Which ones are the readers here? Readers of acetylation. TF2D and switch SNF, right? What protein is the eraser? Yes, the acetylase, RPD3 is the eraser, right? Okay. All right, so that's how you basically clear out the promoter in eukaryotes to start initiation, okay? So in eukaryotes, there's also another coactivator, which is called a mediator. So a mediator complex, what it does is in a gene like this, in mammalian or yeast genes, what you have is you have this transcription start site, you have the promoter, and then you have enhancers that are really far away, okay? So what this mediator complex does is it interacts with the transcription factors that are bound at the enhancer, and it also interacts with the RNA polymerase that is bound at the promoters, thereby mediating that activation of the gene between the enhancers and promoters, forming these loops, okay? So this mediator complex is pretty amazing. It's really well conserved. It's made out of many, many subunits. And if you look at the mediator in yeast and you compare it to the human mediator, you find the same subunits over there. It has certain modules that accomplish different things. And if you look at the structure of the mediator and look with the RNA polymerase, what you can see is that a certain surface of the mediator is making very strong interactions with the RNA polymerase, and the other surfaces of this mediator would be interacting with the transcription factors. Okay? All right. So far, here's just a little summary, right? What you have is sometimes you can have genes in the closed chromatin, and you would have transcription factors. For example, if this gene needs to be transcribed, you can have transcription activators, one or more, typically a lot more, that is going to bind to the promoters and the enhancers. And these transcription factors are going to interact and bring in chromatin coactivators, such as SAGA, right? and these coactivators are going to acetylate the nucleosomes that are nearby, and that's going to lead to removal of the nucleosomes here, and so that you can assemble the pre-initiation complex right here with RNA polymerase so that it can start transcribing. But something curious happens in eukaryotes. For some of the genes, almost half of them, after starting transcription initiation, polymerase pauses, just stops. Okay? So what happens is that you have this initiation complex. There's a protein complex called NELF. It's a negative elongation factor because it negatively regulates elongation. It doesn't let the elongation to happen. What it does is that when the RNA polymerase is released from the promoter, so it's no longer associating with the initiation proteins, it goes in about 1040 base pairs in to the gene, and because it gets the NALF protein interacts with the RNA polymerase and some associating proteins, it stops the polymerase. So you have a little bit of nascent transcript, a little bit of that new RNA made, but you no longer continue. What happens is that then if there is a signal that comes in and activates a protein complex called PTFB, it phosphorylates the RNA polymerase and removes the NALF from the RNA polymerase so that RNA polymerase can be released into elongating. And so the methylation can be added, the RNA can be capped, and the polymerase can continue elongation. Okay? So the RNA polymerase pauses. So what do you guys think would be the function of RNA pol 2 pausing in eukaryotes? Why might you want to actually pause your RNA? Any ideas?
There's like a break, or like a break.
If there's a what?
A break, like if there's a replication protein trace.
You're thinking, yeah, mechanistically, there's replication going on. Maybe you want to just wait or something like that. I don't think that's actually the case, but I don't think there's a lot of research that tested that kind of hypothesis. Were you guys overwhelmed with how many proteins need to come in to the promoter? Oh my God, right? That initiation is going to take some time, right? But sometimes what you want to do is you want to just turn on your gene like this, and you want to do it very fast. So what you do is you want to wait for a signal. And what is thought here is that there are some genes that need to be turned on very quickly, precisely at the right time. So what you do is you get through all that initiation business, right? And have your polymerase standing here just waiting that one signal to remove the NALF and go on and transcribe the gene. Does that make sense? So that's kind of the idea behind this regulatory step. All right, next the polymerase is happy, it's going on elongating, it closes onto the DNA, etc. And it has a pretty good processivity, so it can hang on. And the reason why it has to hang on is some of the genes in humans, for example, are really long. So there's one, the longest gene in humans is 2 megabase long, and the RNA polymerase speed is about 1 to 2 kb per minute. And so it would take 1 day to make one transcript, right? So that RNA polymerase 2 should hold on to that gene for 1 day and continue and finish the transcription. Amazing. All right. So you have RNA pol 2 now starting transcription, but the nucleosomes are not just at the promoters. They're also in front of the RNA polymerase, right? And you need to go plow through them and remove them. And there are elongation factors, these proteins that bind to the elongating RNA polymerase that actually mediates that. So that as the nucleosomes need to be removed, they get removed from the front and then put back put behind the RNA polymerase, so the RNA polymerase can actually continue. So this is actually really important. So when the RNA polymerase passes, you restore the original chromatin state behind the polymerase. Does anyone have an idea why restoring nucleosomes behind the polymerase might actually be important? Why do you want to put back the polymerases behind? Very good. It will prevent the initiation of transcription downstream of the actual transcription start site. And the reason why that occurs is, you guys remember those initiation elements, consensus sequences, how crappy they were? It was like TA, blah, 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 very short, not very specific, right? And by chance, you can actually find those sequences inside the genes as well. But the DNA containing those sequences inside the genes are typically nucleosomal, and they don't contain transcription factor binding sites that are nearby, so they don't get accessible to the initiation complex. But if they're actually, if you don't restore the chromatin structure and you leave these sequences open, and if there is an internal sequence that resembles the Tata box, sometimes by mistake RNA polymerase II can start coming on there and start transcription over there, and then what you will have is you will have incorrect transcription start, right? So you would be starting the transcription in the incorrect size. Does that actually occur? It actually occurs, and it occurs a lot in cancers. So when I said that C-terminal domain that has all these repeats, they get phosphorylated at multiple sites. Those phosphorylations, those modifications serve as binding sites, serves as flags for certain proteins that need to do these nucleosome removal, restoring the chromatin, etc. Okay? Don't worry about all of these proteins. Here, just look at this protein called SET2. What SET2 does is it interacts with the phosphorylated CTD of the elongating RNA polymerase II and travels with it. And what it does is it contains a methyltransferase domain that methylates the histones in the nucleosomes that are behind the RNA polymerase. And that methylation is required for suppressing that internal transcription initiation. Okay? So, in fact, when you look at different cancers in humans and ask, are there mutations that you frequently find that are associated with certain cancers? So these are mutations associated with cancers, and plot some of these mutations at SET2, what you will find is that there are a lot of mutations that you find in cancers that are accumulating at the SET domain. That's the methyltransferase domain. That's the functional domain of this gene. Okay? So why is SET2 mutation actually causing a lot of cancer is not very clear. But if you take those cancer cells that contain these mutations, isolate the RNAs, and look, are there any mistakes that were made? How does the transcriptome means all the transcripts inside the cells of these cancer cells look like compared to normal cells, okay? And researchers actually have done this, and what you would find for a normal gene, what you would find is in the DNA you have an exon 1, intron, exon 2. In normal mRNAs you would find the intron splice and you would find the mature mRNA, right? And in these cells what they find is that they find transcription starting downstream of the actual site, right? So you have shorter exons, right? And interestingly, they also find that in some of the genes, instead of these introns being spliced, the mRNAs actually contain the introns. So the introns are retained. This is called intron retention. And these mRNAs are not going to give rise to normal proteins, right? When you see this guy, so you look at all the genes and you look, a lot of genes have these intron retained. What would your guess be for the function of restoring chromatin behind the RNA polymerase? What is it doing? What must it be doing in addition to suppressing internal transcription? What can rise to this kind of problem? What has to happen for this intron to be retained? Splicing must be problematic, right? Instead of being spliced out, splicing proteins are not doing their job, suggesting that elongating polymerase and that chromatin structure is not only important for repressing transcription internally, it's also important for splicing, right? So there has to be some way that function is required for splicing. And in fact, nowadays people think that the elongating RNA polymerase and the chromatin structure behind that structure is recognized by a lot of the splicing factors to be able to splice the introns in the right place. Okay? All right, so elongating polymerase, it went in, it's going down, everything is good, and the transcription needs to end. So the transcription termination has to occur, and in eukaryotes, the transcription termination is mediated by a bunch of proteins that recognize the C-terminal domain of RNA polymerase. And the RNA polymerase actually pauses again at the transcription end site, and at these sites what you typically find is a stretch of A's. Okay? What does this actually remind you in terms of prokaryotic transcription termination? Do you also find a stretch of A's? Yes? And why was that? What is it?
Just for the row independent.
Row independent termination. So why do you have that stretch of A's? What do?
They don't bind as tightly, so the polymerase attachment to the strand is looser.
Their U to A complementarity is a lot weaker, right? So you have a stretch of A's in the DNA, a stretch of A's in the DNA. So you have a DNA RNA duplex that has that are weaker, so that duplex can be separated. Okay, so the RNA can fall off from the DNA. Does that make sense? Okay. So at that site, it kind of pauses. That pausing now recruits all these termination proteins, and then you get you release the RNA, and the RNA polymerase at the termination site has a typical CTD phosphorylation, which contains low serine 5 and high serine 2. All right. So that's a lot about RNA polymerase 2. The reason why we spent so much time on RNA polymerase 2 is a lot of these protein coding genes, a lot of functional non-coding RNAs are actually transcribed by RNA polymerase 2, but there are other classes of genes that are transcribed by the other classes of RNA polymerases. Okay, and the one that transcribes the rDNA cluster. So this is the rDNA cluster here. What you have is you have these genes that are repeated tandemly many times in the rDNA locus in the genome. They have a single transcription start site, and that transcription start site contains a promoter. And the sequences at the promoter of these RNA genes is different from the sequences that you find at the protein coding gene promoters. Okay? And the reason why these sequences are different is that they are recognized by a different set of initiation factors. So these initiation factors, some of them are named here, are going to bind and they're going to recruit the RNA polymerase 1 so the RNA polymerase 1 can start transcription. So just for convenience, a lot of these transcription initiation factors for RNA polymerase 2 is denoted like TF2B, TF2H, right? Those are the factors that are associating with RNA pol 2. And the ones that are associating with RNA pol 1 are then TIF1A, so it's associating with 1, right? So what you have is you have a separate machinery for the RNA genes. RNA polymerase 3, on the other hand, transcribes a lot of transcribes the tRNA genes, and they also transcribe some non-coding small nucleolar RNA genes. So, for example, here U6 snRNA gene. Some of these small RNAs are also used for splicing, right? In the splicing complexes. But in this RNA pol 3 promoters, you can find different conformations of the promoter. What I mean by that is this. So for a lot of the tRNA genes, what you have is you have promoter elements that again contain sequences that are different from RNA pol 1 and pol 2 promoters. Okay? So these sequences are found in these TF3, are recognized by this protein complex called TF3C. Again, you can see by convention this is associating with RNA polymerase 3, right? TF3. So this is an initiation protein that recognizes these sequences in what we call box A and box B at the tRNA promoters, and then interact with additional initiation factors, then position the RNA polymerase 3 to start transcribing here. And this is the promoter structure for the 5S RNA gene, which is also transcribed by TF3B. And in that case, that gene contains a different sequence element that's at the promoter that's recognized by a different set of initiation factors. And in the snRNA genes, again, what you have is you have a different set of transcription initiation elements that are bound by initiation factors that are going to bring RNA polymerase 3. So just because we spent a lot of time on RNA polymerase 2, don't get the false impression that that's the one that does most of the RNA transcription in the cells. In fact, RNA polymerase 3 probably does the most transcription because it produces a lot more tRNAs. Okay? So it's going to constantly come in and transcribe and transcribe and transcribe all the time. But what RNA polymerase do is it transcribes more genes, a more diverse set of genes than RNA polymerase 3. All right, is it just 1, 2, 3? No. Plants are weird. They contain more. So what they do is they have more specialized RNA polymerases called RNA polymerase 4 and RNA polymerase 5. And these are specialized to transcribe part of the genome that contains and encodes for short interfering RNAs. And you're going to learn more about these in the lecture where you're going to learn post-transcriptional gene regulation, where short interfering RNAs are going to repress genes. Okay? And that's it for today, and I'll see you in a couple of weeks, I think.